Okay, so I'll be talking about some fairly basic stuff because I'm not sure how many people have used datR. So I'll start fairly basic, and then for the detailed stuff, Renee's going to come in later and talk about some more detailed stuff that'll tantalise the people who are more experienced with datR. So basically, I'm going to talk about the data structure that's used by datR, because that's quite important, especially if you want to have a look at the data in any sort of sophisticated way. So the first thing, how is genotype data stored in datR? And it's stored, as it turns out, in a genlight object which has been produced by datR, by the developers of datR. And the reason it's so important is because anyone who's downloaded a data file from Onjay would know that they can be very, very large. And they don't need to be very large because you don't need the full ASCII set to represent the characters that are in these data sets. And so datR uses a much more collapsed alphabet and collapses it right down to a very small file that can be emailed to people, can be read in, really quickly modified, saved. So it's really important that it's in this sort of datR format as a genlight object. The second thing I'll cover is how you examine the contents of a genlight object. So once you've got your data from Dart, or once you've got it from your own SNP calling, you get it into a genlight object, then you want to see, well, what's in there? You want to interrogate it. So I'll talk about how you interrogate it. And then the next thing I'll talk about is how you select subsets of your data. So you might want to, for example, just pick all the males out of your data set, or you might want to eliminate some populations and keep some others, or eliminate some individuals and keep others, or eliminate some loci and keep others. So you might want to subset your data. And then I'll talk about some really basic filtering and the philosophy behind datR, which is basically you do a report on a particular attribute, you then look at the results of that report and decide on a threshold, and then you filter on that threshold. So report, filter, report, filter, report, filter. So it's not like Radiator. I don't know if anyone's used Radiator, where you run Radiator and it runs a whole analysis for you and you put a whole heap of options into it. We've got a different philosophy. It's very particulate. So you run a function that does one thing, and then you daisy chain them all together into whatever it is that you want to do. And then there's some tutorials, and I'll point to those tutorials, how you can learn more. And those tutorials are really straightforward. They're using the best sort of pedagogical techniques that came out of a grant from the Commonwealth Court grant for how to do online teaching. So basically that's it. So if we take a look at the actual genlight object, you find that basically you've got your individuals down the side and you've got your loci across the top, and then you've got your scores in the body of the table. And those scores are 0, 1 and 2. Note this is different from the scoring that Dart uses. So 0 is the homozygous reference state, and 2 is the homozygous alternate state, and 1 is the heterozygote state. So 0, 1 and 2 is a really good reason for doing that, because it simplifies the computations behind the scenes, because you can just sum these things and get the number of or the frequency of the alternate allele just by summing them. So it makes it very easy computationally to have this scoring. And then of course you've got missing data. Oops, yeah, sorry. Talk about that in a sec. So you've got, associated with this data set, you've got 2 data frames in Dart, and they're linked inextricably to the data in a genlight object. So you've got your locus metadata. They're all the attributes of the locus that's being scored, and Dart provides you with a whole suite of attributes automatically in their report. So you've got, for example, you've got your allele ID, your clone ID, allele sequence, you've got the actual SNP state change, SNP position in the sequence tag, you've got the call rate, all the way through to the repav value. And they're all associated with the each locus has these attributes associated with it, and it's in a data frame that's next to the actual data matrix. And it's linked to it in the genlight object. You don't have to worry about that. And then you've got the individual metadata, and some of that comes from Dart. So you've got I guess the service number, you've got the plate number, and you've got the cell associated with that particular individual. That comes from the Dart report. But you might want to put in latitude, longitude, sex. There's a whole suite of other attributes that you might want to put in that are associated with your individuals, and you can put them into the genlight object and then draw upon them as you require. So basically, if you want to access these metadata, your genlight object, which is called GL, has basically, it has this sort of, what's it called? What's it called? It's a slot. Yeah, so you've got this other slot, and in that slot you've got all the locus metrics. And so if you want to refer to them, you can go GL at other $loc metrics $repav, and they'll give you all the repav values for your data set. So you can actually pull these out of the data frame as you require. And the same with the individual metadata. You can pull out the latitude and longitude, again by using GL at other ind metrics instead of loc metrics, and you can pull out all of your ind metrics and then use them in your R programming when you're doing your analyses. You can just draw upon these things. Okay, so that's pretty important to know that that's the way it's structured in the genlight object. And then of course missing data, I'm showing them here as dashes, but they're actually NAs. So anyone that programs in R would know why it's NA. So basically the missing values are NAs. And then we add this other stuff into the genlight object that you probably don't need to know about all of them, but there's these flags in there which indicate that, for example, the call rate's been calculated, and it's in the it's basically in the locus metadata. It's been calculated. It doesn't need to be calculated again. So there's a little flag that says it's been calculated and nothing's been altered that would make those values invalid. If you do something that makes those values invalid, the flag drops, and then next time you go to access them, it recalculates them. So it's just it saves computational time dramatically. And then there's this really natty thing called history, which Bernd wrote. So the genlight object's got the history of every command that was used to get to this point. And so not only can you rerun that history if you wanted to, but you can grab that history and run it on another genlight object. So it's quite a neat little trick. And then there's a verbosity setting, which if you set verbosity to 3, then the output will be quite verbose. If you set it to 2, it'll just say, I'm running this script, I'll finish this script. I'm running this script, I'll finish this script. If you set it to 0, it won't say anything at all. And that's really good if you're batch processing. So you can set the verbosity to 0 and it'll just run and it won't say anything. So that's worth setting. So you just said, GL verbosity 3, and it'll set it to 3 from that point on. And there's other things in there that you probably don't need to know about. So this basically is now, it's a genlight object, but it's a superset, and we're going to call it a datR object. So it's still compatible with the genlight object, so you can use it as a genlight object using all of the ADE genet scripts, but it's a superset which datR draws upon. So that's the way the data's formed, and it's very compact. So it looks like a matrix, but it's not a matrix. Yes? Yeah, the question is, are we talking about how to access these genlight objects? And yes, I'll be talking about that in a minute. So, yeah, so if everyone's sort of clear on that, you've got these genlight objects, and that's what you're accessing when you're doing your analyses. Okay, so how do you interrogate these genlight objects? So the obvious thing to do is just type the name of it. So if you type GL, and that's your genlight object, it'll give you this information. It'll say how many individuals you've got, that's how many genotypes. It'll say how many SNPs you've got, how many SNP loci you've got, in this case 255, it'll say what percentage of the data are missing. So in this case, it's quite substantial, 12.34. So this is a test set. It's got known warts, and one of the known warts is a high missing value rate. And then it gives you other information there that you'll come to understand as you get more sophisticated in your use of datR, but at the moment you don't need it. Then the other thing you can do is, if we just set test set to the GL just for simplicity, so you say GL, put test set in GL, and then you can use these adiagenet accessors to access the genlight object. So you can say, okay, well, n in GL will tell you how many individuals are in there. N lock GL will tell you how many loci. N pop GL will tell you how many populations the individuals have been assigned to. That's the first I'll mention that. So the individuals can be assigned to populations in the genlight object. And N pop GL will say that there are 30 populations in test set. So the individuals are aggregated into 30 populations. And then you can ask for what the pop names are. So you can go pop names GL will give you a list of the names of populations, or you can go in names GL will give you a list of the individuals, or if you want to, you can go loc names GL and it'll give you a massive list of the locus names. I haven't done it here because they're big lists. Or you can go, okay, well, give me a table of the populations labels in GL. So you can go table pop GL and it'll give you a table of all the populations that are in that genlight object and how many individuals are in each population. So, you know, you can interrogate it there really quickly and just see, like, have I read my data incorrectly? That's the first thing I'd be looking for when I do this, is say, you know, are there populations in there that shouldn't be there? Are there some missing? So this is sort of exploratory. And the other thing you can do is you can say, okay, well, I want to look at the actual scores in the genlight object. And you do that by converting it to a matrix of values and then outputting that. So you go as matrix GL. If you do that, you'll get the whole matrix, which will be massive. Or you go as matrix GL, you know, 1 to 7 individuals, 1 to 5 loci, and it'll just give you those. So you can have a look at it and see, you know, what does my data look like? So you can't actually look at the genlight object. If you type GL, you'll get that summary stats table. If you want to look at the actual contents of the genlight object, you have to go as matrix GL, and then you can, you know, you can restrict that in various ways. So you can ask for the first 10 individuals and the first 50 loci, if you like, that sort of thing. You can subset it using R, all the R capacity. Okay, so then, so you can just type GL, or you can use the adiagenet accessors, or you can use our report functions. And these report functions basically give a report that's more sophisticated than, you know, the adiagenet accessors. So you can go, for example, you go set test set to GL, then set the verbosity to 3, and then go GL report call rate GL, and it'll tell you that you've got 255 loci, 250 individuals, and the minimum call rate is 5%, and the maximum call rate is 1, that's 100%, and gives you the median and the mean for the call rate. So the call rate's where you've been successful in calling the SNP, as opposed to not being successful for various reasons. Okay, so it gives you an overall missing rate of 12.34. Okay.
Can I just stop?
Sure.
There seems to be at least some observation of us is you're not really following what Arthur is doing there. You're not following by coding. Is that because you know all that stuff and it's boring, or you got lost how to do that?
I can't log in because it says there's maximum number of student seats as many seats. I'm trying to fixing that.
Yeah, okay. Yeah, okay. Is it how many can't log in because too many students? 1, 2, 3. 3. Okay, sorry, we have to go 3. Okay. No, sorry, we try to fix that. Any other reason why people are not following? Yeah, okay, you know
I'll
that.
slip you
Yeah.
that 50 later.
Okay, maybe.
Am I going,
Let me show.
am I going too fast?
Can I show the cloud and how that works for one of your examples?
Sure.
Yeah. Yeah. Okay. All right.
Not that I'm getting old and inflexible or anything.
Can I ask you a question while we're doing that? So if you go from genetic data down to these three, so if you go from 16 possible diploid genotypes down to 3 categories, 0, 1 and 2, how much information do you actually lose?
So I don't think you lose any information because you've got... Oh yeah, you lose information because if it goes from A to G or G to A, it scores as the same. So
If your homozygous reference is 1, but then you've got 2 is any homozygous alternate, you don't know what the alternate is. It could be 3 options. So I'm just not population.
no, I understand what you're saying. So we're treating it as binary. So you get a mutation at the SNP site, and we're assuming that you're not going to get another mutation at that site, which is probably true except for eucalypts, I guess. In eucalypts, you know, it's probably not true, but in most organisms, you're very unlikely to get a second mutation at the one site. And so basically, we're treating it as if the SNPs are binary, and on that basis, this all makes sense. But if you're treating them as, you know, being A, T, G or C, and all the different combinations, clearly it's not binary, it's multi-allelic. But in practice, it's binary. It's binary for 2 reasons. One is that the SNP is very unlikely to get a mutation at the one site, and the other reason is that in the pipelines that generate the usable SNPs, they'll take out locations that have more than one allele on the assumption that it's probably a duplication, or basically a duplicated site, and they'll carry with them all the multi-allelic sites. But because they're so infrequent, it doesn't cause that much of a distortion. I think that's right, Andre.
Preventing parallels and extracting good data is a process. Yeah. So you need to find the sweet spot because it will vary across the genome and across the population.
Yeah. So basically, you're treating it as a binary character. So you're getting 0, 1 or 2. And if you want to work out what the character state change is, you can look at the actual SNP itself at that locus for that individual, and you can see what the actual change is. And usually I think they score 0 is the most frequent allele, and 2 is the least frequent allele. And so when you do you do the normal histogram, you expect to get a U like that. Instead of getting a U, you get a lopsided U because they've picked the most frequent allele as 0. So you could, if you wanted to, randomise that, but I don't think we've written a script for that.
Yeah, I made it a little bigger. Is that better now? Can people read that better? Yeah? And, yep. No. What? Yeah, I was coming to that. To make this work, you should run this first. That means run. Can you see this? Run everything above. Yeah. So if you start a script not at the very beginning, which I do now, click on this. It runs there. One important setting, which I think is the better one, you can basically say here, chunk output in console, meaning if I run this thing here, it comes up in the console and I have this left and right, yeah? And not inline, because inline means it's in the script, which is a bit annoying because you have to scroll a lot, yeah? So I think this is the best settings, yeah? And so once you clicked all above, you can see it doesn't load datR. Don't do that, Chris. it loads basically database and data, yeah? Because that's the new Dataverse. Okay. And then you can run that as a normal code. You can do a Control-Enter and run that here, and you can see, yeah, that's what Arthur was doing. Test.jl gives you the GL genlight object with all its slot, and then you can do, okay, what is the next one? What about I want to have what? This is weird. Okay, I didn't run that correctly, I think. Sorry. Yep. Phew. Okay.
Yeah,
You never do that running. No, okay.
you saw his deliberate mistake there.
I didn't run this line, and that means GL was a function, so it doesn't matter. Okay. Yeah. And then you can run the code basically here step by step, or you type it yourself, whatever you want, and follow the instructions Arthur is giving, yeah? Does it make sense now? More? Yeah? Anyone still lost? Because honestly, I'm happy to spend 7 more minutes on that, how that works, because that's 90% how our workshop will run. You know, there will be Arthur saying us something, and then there will be code. And quite often you will see only the slides, and you have to run the code yourself, or go back and forth, depends on the presenter, yeah? So please make sure you're able to run the code, and obviously if you can't log in, then we hopefully will save that, yep? Yeah.
Yeah, can
Okay.
you say where the settings are again, Bernd?
Yeah. So here, this little whatever arrow next to the wheel, sorry for that, it's just small. And it says here this kind of thing, and it says junk output in the console and not in line. And if you don't like the images and everything, you can switch them off as well. But I think the images are good because you know where you are in the code, yeah? And I kind of made that here smaller. Whoops, no, where is that now? Doesn't matter. There's the outline here. This can help you as well. You can basically click on here and then you find a different structure, yeah? There's one more thing. I'm not sure if you have a big screen that might help. This whole thing is what I think is really cool. Oh, can I? Green. Kinda. Not the one.
Don't get too fancy.
Yeah, okay, now. Basically, I try to find the link. We have produced, which is called a HTML file from all the sessions, and there was an email, and just can't type the link because I'm not clever enough. I think there's a dot in there, and then it could work. And then there you have all the code as well in there. So if you have space on your screen, you could have this HTML file to the right, and the RStudio to the left, and then you could copy paste it from there. I just try to find, anyone can tell me the link? Emily, what's the actual link then for the, is it in the Zoom tab? So what's wrong with this? It's the other way, is it? It's first GitHub.io and then... Yeah, okay. You find the link somewhere.
I'll put it in the chat soon.
Okay. Cool. Thanks. Can I find it? It doesn't matter. You find it. Okay, and then you could try to use that one. I'm not sure if that works better. It's the same code like this one, so maybe you use the code here, okay? Okay, then back to Arthur, and I'm not sure how to do that here. Yeah.
Yeah.
Okay.
I'm wrong.
And getting to your...
Just get back, that's it. Okay, go up. Oops. That'll do.
Yeah?
Yeah, that's fine. Yeah. Yeah. Okay, so I was trying to get my train of thought back before I was so rudely interrupted, but I'm sure it's necessary. So in terms of following on with me, these are really basic. This is really basic stuff. So if you can't follow on or keep up, there are tutorials I'm going to give you at the end where it's all there blow by blow. And so you can work through the tutorials, or you can work through the online tutorial that Emily's put together. Or you can just copy off the screen there, just jot down off the screen there and run it later, or you can run it now. So I hope you can run it now. So I was under the impression everyone had the instructions for getting on the cloud before we started. So anyway, it's obviously going to take a little bit of getting used to. It's challenging, especially if people are being excluded because there's too many students online. Okay, so let's just jump forward. So you've got these report functions. So you've got this geo report call rate, which gives you some statistics about the call rate. That's the success in calling a SNPs. So they might not be successful in being called because you've used low coverage. And so the read depth is really low. And so certain sections of the genome are completely missed. And so the SNP won't be called for that individual because the read depth is so low. If you're using full coverage, your read depth is about 22, and so the chance of you completely missing a SNP is very low, but you still get missing data because of a mutation at one or either of the restriction enzyme sites. So it's a true null allele. So the missing data is a mixture of SNPs that have been missed because you haven't had deep enough read depth, and SNPs that are missed because of mutations in one or both of the restriction enzyme sites. Okay, so there's two types of sources for missing data. One type is random, that's the low coverage or low read depth source, and the other source is not random at all because those mutations can be inherited. And so a whole population may have a null allele at a particular locus. So it's not random at all, it's being inherited like any other character. So that all comes into play later on. Okay, now the other thing it gives you is this really nice graph which shows you basically the distribution of the call rate across the loci. So some loci have almost zero individuals called, and others have 100% of individuals called. And so here you've got a really nice graph to decide, okay, I'm going to filter my data on call rate. What am I going to use for a threshold? Well, you might decide to just pick that little gap there and filter out the call rate for anything below 0.85. So this graph's really useful because it tells you basically what the call rate is across your loci and how much data you're going to lose if you filter out on a particular threshold. So you might decide, okay, I'm going to filter out all loci that are below that particular threshold of say 0.085. So anything that's below 85% called, I'm going to filter that out of my data set because a lot of analysis is very sensitive to missing data. All right, then you can also use the call rate to report on individuals. So that I just did the report a call rate on loci, but you can report on the individuals as well. So here's a call rate based on individuals, and again you can see there are some recalcitrant individuals there in the sense that some of these have a very low call rate. So for whatever reason, maybe the sample was corrupted or maybe it was left out in the sun or something, and so you end up with a low call rate for those individuals. So you might decide to eliminate individuals that are below 0.85. So you normally do a call rate of 0.90 for loci, and 0.85 is probably a good ballpark figure for individuals. You don't want to over-filter because you're going to cause distortions in your data if you over-filter. So Renee will talk about this. You need to take an experimental approach. You need to play with your data. You need to say, okay, well, if I filter at 0.95, and I get this result, what happens if I filter at 0.9? Does the result change? Or 0.85 or 0.8? And then filter as little as you need to in order to avoid distortions in your data. I think Andre would agree with that quite strongly. So again, you might decide you're going to filter out on a threshold of 0.85 with the individuals, and it gives you the list of the 20 individuals with the lowest call rate, so you can look down that list and see, okay, who's going to go, and make your judgments as to whether those animals are really valuable and you don't want them to be filtered out, or race upstairs to DART and say, this animal's really valuable, can we run it again? In our case, you race upstairs. So it tells you who will be lost, which I think is really important, because you can then look those animals up and say, oh, gee, I don't want to lose that animal. And then you can report on reproducibility. Just as another example, I think you probably realise that DART, one of the great things about DART is it runs your samples, it runs technical replicates on your samples. And I was going to make a joke about that, run it at 10%. But it's at least 30% and can be, and probably usually 60% is my understanding. It's pretty high. What's your...
So it varies depending on the size of experiment. If we have, let's say, 1,000 animals, we would use 10%. If we have 20 animals or 50, we do full replication. Yeah? So we try to get a statistically significant number of replicated libraries to determine reproducibility and other statistics. So we adjust to the size of the experiment, so there's no fixed number.
Yeah, so there's no fixed number, but basically if you do the report on reproducibility, it'll tell you how many you're going to lose if you pick a particular threshold. So if you pick a threshold of 0.9, you're going to lose 9.8% of your data. And then of course it does the graph as well, and so there's a reproducibility, and obviously most of your loci have 100% reproducibility, and you might choose to filter below 0.995. Anything below 0.995 you're going to take out. And again, you be a bit careful not to overfilter, because you can bring in ascertainment biases if you overfilter. So you wouldn't filter on 1. But you might filter on 0.99 and take out those loci that have low reproducibility for whatever reason. Okay, so you filter them out, and you get the gist, I think. And so there's a whole stack of these matched filters. So there's a filter call rate, you've got reproducibility, you can report on secondaries, that's how many sequence tags have more than one SNP in them. So if sequence tags have three SNPs in the sequence tag, you get 3 lines in your data set, 1 for each SNP. And so obviously they're linked, strongly linked, and so you might want to think about how you're going to handle secondaries. You can get rid of monomorphs, so if a particular locus has the same score across all individuals in your data set, you can say no information, throw it out. There's an interesting one, you can get to report on overhang. So does anyone know what overhang is? So obviously haven't encountered this before. So when DART trim the sequences, they trim the adapters off. Every now and again, the true sequence will match the adapter. And so it'll trim off a bit of the end of the sequence tag, which might happen to have a SNP in it. So you end up with an invariant sequence tag that's been scored for a SNP. And so that matters if you're going to concatenate your sequences for a phylogeny, for example, because you're just adding in an invariant bit of sequence in there unnecessarily. So you might want to report on the overhang. You can report on the Hamming distance. So I know that Steve Dinellan, with his squid study down in South Australia, they did a report on the Hamming distance and decided that they'd filter on a higher Hamming distance than DART did routinely. So you might want to do that. So that's how close your loci are to each other in terms of how similar their sequence tags are. So you might want to do that. Overshoot and overhang are the same, I think. They're probably cocked up there. And then you can report on a loc metric. So if you've got a locus metric, any locus metric, you can do a report on that and get a histogram of that particular locus metric, which is quite valuable if you want to use some of the things like avpic, for example.
Can you say to the people, to everyone, that the problem with the number of maximum members has been solved? So
Oh, okay.
if they can now log in again, please?
Okay, the problem of the maximum number of participants that have logged on to the system has been solved. So if you're having trouble before, have another go and see if you can get on the system. Apologies for whatever happened there. Anyway, these are all matched. These are all matched reports in the sense that each report has a matched filter function. So each one of these has a matched filter function. There are a lot of other reports you can do. So you can report on linkage disequilibrium, heterozygosity. There's a whole stack of other reports. I'm just talking about the ones that you use in early manipulation of your data. Okay, so and I've got a running log of what I'm doing over the far side there if you need it. And then you can subset your data. So if you want to, for example, take some individuals out. So you do a PCA or something, you find this really oddball individual and you think, you know, what's happened there? It's probably a misidentification. You don't know what it is, but it's certainly going to mess up your subsequent analysis, so you give it the flick. So you can just go gl drop ind and give it the individual name and it'll throw it out. Or you might find you've got a couple of records that are actually the same individual somehow got in there twice, so you want to take one of them out, that sort of thing. And of course you can do the same with, you can keep a list of loci, or you can drop a list of loci, you can keep a list of populations or drop a list of populations. You can merge populations into one population. You can subsample individuals with or without replacement. So you might say, okay, genlight object, I want to try this new snapper program that's in Beast, Beauty and the Beast, right? I want to try that. Like you know it's going to take 100 days to run if you use your whole data set, so you might just subsample the individuals, subsample the loci with or without replacement, try it out with snapper, get it all working, and then waste 100 days with the full data set to get your outcome. So the subsampling scripts are pretty important. So there's quite a few of these, additional ones of these for subsetting that are in the tutorial, interrogating and manipulating data, which I've uploaded onto the site, but you can also pull it down from the website. And it goes through blow by blow, and it's a tutorial, so you can work through it and work out how to do things, which I think is quite useful. Now recall that you have these report functions, right? And for each of those report functions there's a matched filtering function. So you can, for example, filter on call rate. And of course, when you filter on call rate, you've got to put in a new genlight object or overwrite your genlight object with it. So you can filter on call rate, filter on reproducibility, filter on secondaries, filter on read depth. I didn't mention that one before, so you can report the read depth. And you filter on read depth because, you know, the standard read depth, I think, is about 5x. Yeah, about 5x. You might say, okay, well, heterozygotes are really important to me. You know, I'm going to look at sex linkage, or I'm going to look at something where calling a heterozygote is really important. So you might push if you've got full coverage, you might push the read depth up to 12 so that you're really certain of your het calls. Whereas if you're down at 5x, you know, there's an uncertainty around the het calls that you might not find acceptable. So you might want to play with that as well, filtering on your read depth. You can filter on your Hamming, overshoot. You can that should be filtered at the bottom. You can filter on a loc metric, so you can say, okay, I want to filter on avpic, average polymorphic information content. You might want to filter on polymorphic information content. So you're only after, say you're doing a new hybrids analysis or something, and you know you can only use 200 loci, so you might want to filter heavily on avpic and pick the loci, the most variable across individuals, and then run your new hybrids on that. We're working on getting around that limit of 200, but there's only so many days in the hours in the day. Okay, so I think you should be at least getting an overview of this. And of course, Renee's going to talk in more detail about filtering, because there are nuances around filtering that she's going to cover that I haven't covered. And then, of course, here's another example, if you recall, with the call rate. You can also, as I said, filter on individuals, for example. And when you do the filter on the individuals with call rate, it gives you a before and after graph. So you can see what it was, and you can see what it is after the filtering. And that's really useful. So you use a report to work out your threshold, but when you do filter it, it gives you a before after, so you can see the impact of your filtering on your data. And all of the filter functions do that. So it's quite useful. And it also tells you not only who has been discarded when you filter, it tells you what population they're from. And so that can be really important. So here, you know, it's filtering out individuals from the Cooper Creek in Avon Waterhole. For some reason, those animals didn't pass the mustard with the individual call rate. And so it's filtering them out. If they're really important, then I would go to Onjo and say, oh, there's a problem with these particular individuals, can we run them again? And then combine the service. But they'd have to be particularly important because that's quite a costly exercise. Okay, does everyone understand how this is working? It's pretty basic. I hope I'm not boring the people who are usually using the package a lot. But anyway, then you can do things like a quick graphical evaluation. So you can use this smear plot function. So up the top there, you've got a GL smear plot run before you filtered on call rate. And you can see there, there's a lot of loci. So you've got the individuals up the side and you've got the loci across the bottom. You can see there's a lot of loci that are white. So white's missing values. And so you can see there's an awful lot of loci that have got a problem with a call rate. Just straight away, just looking at that graph, you can see it. You can also see some individuals there that have a problem with call rate. So those individuals there have a problem with call rate on those populations, and same over here. So you can see straight away that, you know, what the impact of call rate's going to have, and then when you run your filters, you end up with this. So clearly it's fixed the locus problem, but you've still got some individuals that have got a low call rate on some loci, but not low enough to have been eliminated by your filtering. So they're still in the data set. Okay, so it's just a way of just playing with your data, doing the filtering, having a look at what impact it's had, using this smear plot, which is just straight out of AUGenet, or whatever it's called. A lot of data is just a wrapper around other people's scripts, but it makes them user-friendly. So you don't need any R program, you use datR, whereas some of these other packages you do. So we just put wrappers around them and we acknowledge it in the script. Okay, so the basic filtering that you can do, again, there's another guide, a guide to basic filtering. And again, if you work through that, preferably with your own data set, because you know your own data set. So work through the tutorial with your own data set and just play with it and see what different thresholds do, etc., etc. Before you launch into a major analysis, as Renee will say. Okay, so here are the tutorials. There's an RStudio refresher for people who don't know so much about RStudio like me. There's how Dart stores your data, there's interrogating and manipulating data, there's installing datR, and there's a guide to basic filtering. And you'll see as we go through the talks that there'll be other tutorials like this that you can draw upon. How many people have used these tutorials? Has anyone used them? Quite a few. Did you find them useful, or are they just too slow? They're okay? Yeah? All right. Okay, well, I think I'm done there. Is there any questions? Yep.
Thank you. You mentioned that you wouldn't filter on a reproducibility of 1. I haven't heard that before. Could you clarify why you wouldn't use 1?
Yeah, so I guess the reason you wouldn't use 1, and Andre can probably elaborate on this, is that the heavier you filter, the more likely you are to introduce some sort of ascertainment bias. And so the idea is don't filter more than you have to. So you would play with the filtering. So you might filter with 1, see what happened, and then try filtering with 0.99 and see if it didn't make any difference to the outcome of your analysis, and then filter as least as you need to so that you don't introduce some sort of ascertainment bias. Maybe, so this is the expert.
Not an expert, but definitely something that Arthur mentioned in passing about read depth, yes, and that's relevant to your question because it will be very costly to run an average of 50 or 100 read depth where we have absolutely no problem of any heads not being properly called. Yes? But in the interest of financially reasonable solution, even if I say that sequencing is getting cheaper and we're actually, for the same price, we'll be giving more sequence very soon, that's our plan, that in the older data sets, that definitely you'll be hammering at those markers that are high in heterozygosity and therefore creating a little bit of ascertainment bias just because of that. Yeah? So it may be still a very useful marker that may be either discriminating or associated with adaptation trait, whatever. Area that we recommend super strict filtering would be either very high resolution genetic mapping where any mistake in calling will create map distortion, and there will be a few other applications, maybe some close relatedness analysis where mistakes in calling would have big impact. A lot of applications in ecology, if you drop down to 0.99 as that to recommend, or even a bit lower that I often do, you will not harm your output, but you may lose something that may be precious. Thank you.
Yeah, he spanked me over this, so that's why I'm sensitive about it. Yeah, okay. Yeah. All right, so are there no questions online at all? So hopefully the online people, if you could not behave like an undergraduate class before COVID, that'd be really good. Speak up.
All right, so we've got one question asking, how do you know which loci are useful or not? And they've got tissue samples and scat samples.
So which loci are useful and which aren't depends on the problem that you're addressing. So obviously if you're looking looking at structure across a landscape, you don't want to look at loci that have a low pick, low polymorphic information content. If you want to filter your data set down to a size that's manageable by filtering out loci to get it down to that size, then you would filter on pick if you're interested in that sort of thing. If you're interested in something like phylogenetics, then obviously autapomorphies have a low information content. So you might want to do a filter that takes out autapomorphies at the population level, and so you could do that. So it really does depend on the analysis that you're going to do. If you're going to do a PCA, most people don't realise this, it hates missing data. So really a classical PCA can't be run with any missing data. And so you would want to filter out the loci, you want to filter more stringently on call rate before you do a PCA, because the methods for imputing the missing data, which are hidden in the algorithms, can cause distortions. And so under those circumstances, you want to filter out loci really more heavily than you would otherwise do on call rate. So yeah, it depends, horses for courses. Yeah.
And what happens if you don't have full coverage with any of the loci? And I guess, like, how do you know which samples to keep?
What happens if you don't have full coverage? So it's a cost trade-off, isn't it? So if you have low coverage, then you're going to end up with less loci after you do all the filtering. So instead of having, say, 15,000 loci, you might end up with 5,000 loci that are reliable and relevant to your questions. So that's the first thing that happens. And the second thing that happens is the reliability of the heterozygote calling is low and irrecoverable, because you can't push the read depth up to get the heterozygosity rates really accurate, because you run out of read depth. So I don't know, in low coverage your read depth might be 7x. Would that be right, Andre? Be about 7x. And so if you need to push it up to 12x to get the real accuracy on your hets, obviously you don't have the room to move. So yeah, there are consequences for going with low depth, but again, it depends on the question you're trying to address and how many loci you need to address it. So I wouldn't be recommending everyone go high coverage for every question. In fact, Pete, you run with half coverage for most of your questions, right? So half coverage for most questions is probably adequate. Low coverage might be adequate for maybe, I guess, for identifying individuals. Let's see, maybe for individual assignment. Yeah. All right.
Thanks very much, Arthur. Oh, it's so loud. So I would normally check my data set post-filtering with any particular step with the actual analysis to see if it changes the result, rather than just looking at the plot, for example. I mean, maybe not for the call rate, but for some of those other ones to check what their effect is on the... would you recommend that? Or?
Yeah, definitely.
And so that's a two-part question, sorry, because I wonder if maybe this is in Renee's session, but any tips to actually keep track of what you've tested and, what the outcome of that was would be great as well.
Yeah, so history, you can do the...
Yeah, I know.
You can do all these things and then you can check on the history.
So is that only relevant for a particular object, or is that like for all objects?
No, it's stored in the particular object.
Yeah.
Yeah.
So you might use different objects.
You just have to keep your script, I guess.
Yeah.
And yeah, in terms of the first question, you definitely want to experiment, as Renee will explain. So leave it for her.
Yeah, so Renee, yeah.
So just a question related to that previous question. So would you suggest doing different filtering steps for different analysis?
I'll leave that for Renee. That's her favourite topic. So that leads into Renee, I think. So Renee, do you want to?
No, Renee is coming at the beginning of the next session, because now it's time to go to have some dinner.
Oh, okay. All right. Well, did I blow it out? Oh, maybe I did.
Fine.
Sorry. Yeah. All right. So, so we're going to have a break for dinner, and then Renee will answer that question. That's what her topic's on. So that'll be really good. Yeah. All right. And if there's no other questions, so you can catch up with me any time this week and ask questions, or if you want help and I'm not in the session, we can go over and sit over there and work through things. Whatever you like. Thank you very much.